Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you another really fascinating guest involved in science, technology, medicine that's creating a better tomorrow for many patients out there. Today, on the show, we have the honor of being joined by Dr. Frank Bedu-Addo, who has served as Director, President, and Chief Executive Officer of PDS Biotech. Since its inception, they are a biotech company. They're focused on transforming how the immune system targets and fights disease by developing a new generation of targeted cancer treatments. Dr. Bedu-Addo is a veteran biotech executive with experience not just starting, but growing various biotech organizations, has been responsible for the development and implementation of operational and drug development strategies, as well as supervising and managing both large and small biotech companies. Dr. Bedu-Addo was a founding and senior executive at KBI BioPharma. There as VP of Drug Development, he oversaw all their business and drug development operations. And before KBI, he successfully started to manage Cardinal Health's East Coast biotech drug development operations. And prior to Cardinal Health, he was associate director at Akzo-Nobel, also senior scientist at Elan, Liposome company, as well as principal scientist at Schering-Plough, contributing to the development of numerous drugs, including those in the antiviral and cancer portfolios. Dr. Bedu-Addo attained his master's in chemical engineering and Ph.D. in pharmaceutics from the University of Pittsburgh. A lot to get into today. Looking forward to having him. Dr. Frank Bedu-Addo, thank you so much for taking time to come on the show.
Well, Ira, thank you very much for the introduction. And I know that this is a chance to introduce you and your listeners to PDS Biotechnology and our work in targeted cancer immunotherapy. And so I really appreciate that opportunity. Before we get started, I'll ask you, how much do you know today about PDS Biotechnology and our Versamune immunotherapy platform?
Well, so I didn't, if I could hang in there with you, and I didn't want to sound stupid. I did a decent amount of research, but what I would love to do is start off, because I know your portfolio, unlike several that we've profiled on the show in the immuno-onc space, has a couple different arms to it. Before we get into it, lay the groundwork a little bit, because we've sort of gone broadly in immuno-oncology in the past in terms of CAR-T, in terms of antibody drug conjugates and so forth, neoantigens. You're bringing a lot of this together. Talk a little bit because there's two platforms here. One that is an antibody drug conjugate that is focused on impacting how cells protect themselves, cancer cells, from the immune system, and then a neoantigen platform. Talk a little bit about both of these, because I think they're kind of important, and then we'll go into detail of both.
Yes, no, so I'd be very happy to do that. So at PDS Biotechnology, what we're doing here is we have a really sharp focus, as you can see, on advancing our targeted immunotherapy platforms to address significant unmet needs in treating cancer, right? So you did mention a number of different approaches. So when we look at the need and opportunity in oncology today and how an immunotherapy could potentially fit in and address some of the limitations in cancer treatment today, such as patient survival, we don't have to look much further than the CAR-T cell approach. The clinical successes of the CAR-T approach have proven a very fundamental principle in oncology, which is if you can generate a sufficient quantity of potent tumor-targeting T cells, which are a component of our white blood cells, we can induce significant anti-clinical benefit. Right, so that principle has been proven with CAR-T. However, despite these successes with CAR-T, CAR T cell approaches haven't been widely applied to cancer treatment today because they have a significant number of major challenges, right? When we look at the CAR T, the high cost and complexity of these approaches, right? Manufacturing these personalized cellular therapies is really labor intensive. It's highly expensive and very often not logistically feasible at scale. These approaches have also run into some significant toxicity issues. Right, we have with these CAR T cell approaches very significant immune-related adverse events. We've seen cytokine release syndrome in several patients. We've seen neurotoxicity and levels of toxicity that require the patients to be hospitalized for long periods just to manage these side effects. Right, these CAR T approaches also haven't had very much success in the solid tumors, even though they've had a lot of success in the blood cancers such as lymphoma. Efficacy in the solid tumors, where these tumors have the ability to generate an immunosuppressive environment, these approaches haven't been very successful. And the other limitation of the CAR T approach has been the memory T cell approach. So our immune systems have the ability to generate a memory T cell response that allows the immune system to recognize the target over a sustained period of time and therefore generate a more durable anti-disease immune response. And that has been one of the key limitations of the CAR T cell approach. And so despite that important proof of concept, these approaches have still created a clear need for immunotherapy, where if we can deliver an immunotherapy that has comparable T cell potency, but without the limitations of these cellular approaches, there is significant potential to significantly impact treatment of cancer. Now, when we talk about these non-cellular approaches, the one that clearly comes to mind are the cancer vaccines, right? The cancer vaccines have also fallen short because of one major limitation, which is insufficient ability to induce adequate numbers of the right type of tumor targeting T cell. And with the cancer vaccines, one of the key limitations that we know today, why we see this limitation is, of course, the inability to activate the right immunological signaling pathways while simultaneously presenting that tumor-specific protein, also called the antigen, to the immune system. So bringing those two together effectively has been one of the key limitations of the cancer vaccine approach. Another key limitation that has also impacted the ability to induce these T cells is gaining access into the lymph nodes. So our lymph nodes are really the T cell hub of the human body. That's where the T cell priming or activation and where the T cells are trained to recognize the specific disease occurs, right within that lymph node. And the limited ability of cancer vaccines to effectively access the lymph nodes and also to activate the right immunological signaling processes within the lymph node have significantly impacted the ability of these cancer vaccines to generate the right types of powerful T cell responses that would be necessary. With these cancer vaccines, we've also had some limitations in terms of the manufacturing. So encapsulation of antigens, vectorization, for example, all these which impact the logistics and also the commercial feasibility and also the regulatory pathways have all impacted the ability of cancer vaccines to be successfully commercialized. And we haven't even talked about the limited genetic restrictions, right? We've seen very often with a cancer vaccine, you'll find activity or good activity in a very small genetic subpopulation of patients, but we don't see that efficacy across a broad range of genetic diversity. That's also one of the big limitations of cancer vaccines. And so even though these proof of concepts or these principles have been successfully demonstrated, we still haven't been able to generate the ideal immunotherapy that's devoid of these limitations. And that's where we believe the Versamune technology platform comes into play, right? So with the Versamune platform today, we have run 3 independent or reported results from 3 independently run phase 2 human clinical trials in different indications. So we've run a trial in cervical cancer that was led and run by MD Anderson Cancer Center. We've also had a trial led and run by the National Cancer Institute independently in all types of HPV-related cancer. So these would be advanced cervical, anal, vaginal, vulvar, head and neck cancers as a basket trial. And we've also run a global multi-center phase 2 trial in recurrent metastatic head and neck cancer. In each of these 3 trials, we have reported the longest median overall survival reported to date by far in each of these 3 indications. And so what this tells us is that this next generation Versamune technology platform has the potential to overcome some of the key limitations of both the cellular approaches like the CAR-T and the limitations of the cancer vaccine approach, right? So with this technology, it also allows us to tailor the immunotherapy to match the specific tumor type that we're seeking to treat. It's a very, very simple lipid-based approach that is able to activate the right immunological signaling processes, gain access into the lymph nodes with a simple subcutaneous injection, either in the upper arm or the upper thigh, and also to alter the tumor's microenvironments. As I mentioned, with the solid tumors, very often we'll run into a situation where these tumors are able to generate or create an immunosuppressive environment, and the ability to overcome that environment and to convert it into an immune submissive or immune permissive environment allows these T cells to be much more effective. So that's really our Versamune technology platform. Really, the platform really activates the immune system to recognize the specific protein typically related to a specific tumor that we would like to activate. The first indication we're looking at is head and neck cancer, right? And we can go into some of these in a little bit more detail. And that's it. We've moved into a phase 3 human clinical trial. Many people don't realize. Ira, go ahead. You had a question.
Yeah, no. Before we get into the clinical work, I just want to just go a little further, just so the audience because understands there's really a sort of a 1-2 punch here. One, the neoantigen presenting basket, which incorporates the antigens, the interesting liposomal delivery system, plus something known as PDS 0180C. This is a antibody drug conjugate that specifically targets IL-12. And this is what you're getting in. So, the antigens go to the lymph nodes and present. The IL-12 is going to tumor microenvironment to say, hey, shut down, this excessive immune response against what's coming. Say a few more words about that, because you're a pharmaceutical, you have a PhD in pharmaceutics, and behind that in itself, both of these have very interesting pharmaceutics story to them. So just go a little further there. I think it's important.
No, great question. So with the Versamune technology, you mentioned that this is a liposomal nanoparticle, right? And so this is made up of a lipid. And what's unique about this lipid is the fact that this lipid is the first time that immune activation has been shown specific to a lipid. So this is an immune activating lipid. We make it into a liposomal nanoparticle simply to achieve the goal of efficient uptake by the immune system. Right. We know that the immune system is built to detect and take up particulate matter, right? And so this approach is really just to get the immune system to take up that lipid. We've also made our manufacturing very simple by overcoming the need to encapsulate these protein or peptide antigens into the nanoparticle. So we've come up with a proprietary approach to make these spontaneously formed particles themselves. So now we have the liposomal nanoparticle and we have the independent antigen particles, right? The proteins or peptides which form these particles. And so both of these form spontaneously. And so we have a very straightforward manufacturing. These particles form spontaneously, they're sterilized and vialed, right? And so these successfully scaled up this process. And so what this does, as you mentioned, is this will take this combination of the Versamune nanoparticle and the tumor-specific protein or peptide antigen that's injected into the patient by simple subcutaneous injection. What that nanoparticle then does is it activates those immune cells in the skin area. right? And by those immune cells becoming activated and maturing, their natural process is to migrate into the lymph node. That's the way the immune system is built, right? And so the immunotherapy gains really effective access into the lymph nodes, right? So one of the key, you may be asking, well, why does it go? Why are they taken up so efficiently and going to the lymph node versus other immunotherapies? This is a positively charged lipid. Our cells are negatively charged.
Okay.
So it's actually sticky. So when you give it in the injection site, it forms a depot by these positive charges getting stuck to our negatively charged cells. And by forming that depot, it gives the immune system enough time to come and pick up these particles. Because of the fact that it activates the right immunological signals, it actually sends signals to those immune cells to come to the skin area, to the injection site, to pick up these nanoparticles. So you'll see some redness and swelling at the injection site. That's a sign that the immune system has been successfully activated. And so those immune cells come pick up the nanoparticles, they get activated, and then they go into the lymph nodes. And what we've shown is that when it gets into the lymph nodes, it also activates the right signals to the T cells, to signal T cells to come into the lymph node. Because the key thing is we have to be able to recruit the soldiers, right? The T cells are the soldiers. So that's an important message to be able to tell these soldiers, hey, come into the recruitment center, right? We've got to train you. So those T cells come into the lymph nodes, and that's where those nanoparticles can then present that tumor-specific protein or peptide to those T cells. And that's the process by which the T cells get trained to say, hey, this is a foreign agent. We've got to invade the body, seek out and kill any cell in the body that contains this foreign agent, which should be the cancer cells, right? And by activating the right immunological signals, it also activates the T cells. So it arms the army, right? So recruit the army, train the army, and then send them out to go seek the enemy and kill the enemy, which should be the cancer cells. Now, you mentioned that very important point, which is the IL-12 ADC. So we talked about immune suppression. So the enemy, which is the tumor, has built up its own defenses, right? And that's how cancer survives and thrives in the body, by evading the immune system or tricking the immune system to believe that it's a normal cell and shouldn't be attacked, right? IL-12 has been out there for a couple of decades. People have tried to utilize IL-12 to activate the immune system. However, IL-12 has been extremely toxic. It's a very potent cytokine, and it has been administered by IV systemically in the blood. It's a T cell activator, right? And so just thinking about T cells and how they work, they have to track to the tumors. If you have IL-12 circulating in the blood, the patient's blood, right, it's circulating in the area where it is not really needed. Where it's needed is within the tumor, right, to activate T cell in the tumor. What our antibody drug conjugate does is it's conjugated to the IL-12 is conjugated or fused rather to an antibody that binds to free DNA. In the body, where do we find free DNA? In the necrotic tumor cells. And therefore, this antibody takes the IL-12 into the tumor's microenvironment rather than having it hang around and create toxicity in the circulating blood, right? And so now we have these two. So this is the study actually that the National Cancer Institute performed. So if you look at our study in head and neck cancer, where the standard of care today is Keytruda, Keytruda plus chemotherapy, or the EGFR inhibitor cetuximab plus chemotherapy. Whichever way you go in first-line recurrent metastatic head and neck cancer, the median overall survival is about 12 months, right? What this means is that if you are diagnosed with recurrent metastatic head and neck cancer, you have a 50% probability of living 12 months. That's the state of the art today. However, when in our phase 2 clinical trial, when we combined our Versamune HPV, so PDS-0101, so that's Versamune plus HPV-16, which is the tumors are positive for HPV-16.
Okay.
What we have reported is a median overall survival of 30 months. And so what that means is that if you were diagnosed with this disease and you went onto the PDS clinical trial, you had a 50% chance of living 30 months or more. We have several patients who are actually approaching 4 years of survival today. Several have passed 3 years of survival today. So we see the significant potential in increasing survival of these patients. Over 20% of the patients had complete or near complete tumor shrinkage, 90 to 100% tumor shrinkage. And so that is just combining our PDS-0101 with the checkpoint inhibitor.
Right.
What the National Cancer Institute did is they said, okay, now let's add your IL-12 antibody drug conjugate. That IL-12 targets the patient's tumors. It induces inflammation in the tumors, so makes the tumors more amenable to T cell attack, but can also further activate those T cells once they enter into the tumor. So two things are necessary. Getting IL-12 into the tumor is one thing, but also having the immunotherapy that can generate those tumor-infiltrating T cells is also critical for those to work, because both have to get into the patient's tumor, right? So having the IL-12 without that T cell machine doesn't really help you, right? And that's where our Versamune becomes very important. And so what the National Cancer Institute showed, they did this in two groups of patients. One group of patients were those who had failed prior treatment and would traditionally be going on to checkpoint inhibitor therapy, right? These are the patients whose median survival, average survival is about 12 months, right? In these patients with our Versamune, we went from 12 months to 30 months. By adding the IL-12 in the National Cancer Institute study, at 42 months, they had still not yet reached the median overall survival of these patients. Right, so we've gone from 12 months now to 30 months with our immunotherapy, adding the IL-12 antibody drug conjugate, 42 months and counting. But the other thing they did was they said, let's take patients who have also failed checkpoint inhibitor therapy. Now, these patients, when they are HPV positive, only have 3 to 4 months left to survive. So their median survival is about 3.4 months, and this is published. Right. And so with this combination, what they showed was that in their study, they were seeing a median overall survival of these patients of 17 to 19 months, right? So we've gone from 3 to 4 months when they failed checkpoint inhibitor therapy to now almost 2 years of survival. And so what all this has shown us, again, really emphasizing that basic concept that if you can generate the right type of tumor targeting T cells, and you can overcome the tumor's immunosuppressive mechanisms, we have really significant potential to really advance the treatment of some of these really debilitating cancers and really extend patient survival quite significantly.
No, it's an extremely elegant package, again, that merges, as you were saying, all the pieces of antigen presentation in terms of the antibody drug conjugates, interesting pharmacokinetics and pharmaceutics behind it all. So, and just to emphasize for the audience, we're talking about the United States alone. There's close to 100 million people that are infected with HPV, even though we do have a vaccine out there. So there's a huge potential market, not just in the US, but overseas as well for the potential HPV cancers that trickle down from this. So, that's. You mentioned the antigen, in that case, something like HPV-16. Could you just say what is? Could you just say a couple words what that is? Is that related to the virus itself? Is it specifically? Just personally curious what that is.
No, you do raise a very important point that most people don't actually realize. So with head and neck cancer, for example, there are two types of head and neck cancer. We have the head and neck cancers caused by tobacco and smoking, or tobacco and alcohol, rather. And we have the head and neck cancers that are actually caused by a viral infection. Yeah. In the United States and Europe today, the most prevalent or the most common type of head and neck cancers are those caused by the viral infection. And it's actually very often been described as a silent epidemic because of the rapidly increasing number of incidences of these virally driven head and neck cancers in the male population that no one is really talking about, right? And so the human papillomavirus is the most prevalent sexually transmitted agent worldwide. It's extremely prevalent among the population. Now, there are over 100 different types of HPV. And when I say HPV, I'm referring to the human papillomavirus.
Right.
However, there is one type among the over 100, the HPV 16, right? It's called HPV type 16. And the HPV type 16, based upon its biology, has figured out a way to shut down the immune system's ability to detect it, right? And what HPV 16 then does is it integrates some of its proteins into the host DNA.
Mm-hmm.
It transforms the cells into cancer cells, right? And so HPV type 16, also known as HPV 16, is by far the most oncogenic type of HPV. And HPV 16 head and neck cancer today is the most prevalent type of head and neck cancer in the United States and Europe today, and growing very rapidly. There are a couple of reports that have even suggested that by the year 2030, this will be increasing at the rate of about 30% a year. Right, so a very dramatically increasing cancer. And head and neck cancer, unfortunately, is a very debilitating cancer, right? The suicide rates in head and neck cancer is about 3 times the average rate in the United States, right? And these patients, unfortunately, due to even though they have very different biology from the traditional head and neck cancers, right? They have very different pathology. There's still no HPV 16 targeted immunotherapy today, right? These are treated the same way as the traditional alcohol and tobacco related cancers are treated. And we've ever had the question, there's an HPV vaccine out there today. Why are we seeing these dramatic increases in HPV 16 specific head and neck cancer? And there are a number of reasons for that. Right, the uptake of the vaccines hasn't been as high as you would expect, especially in the male population. Right, with the very high prevalence of HPV infection, the other thing is this disease progresses very slowly from infection to cancer. In some cases, it takes a couple of decades, 20 to 30 years, to go from infection to cancer. And the current HPV preventive vaccines have to be given before the patient is actually exposed to the virus, because once the patient is infected with the virus, they are completely ineffective, right? And so what's been shown, and this was published in the medical journal Lancet 2 years ago, that this increase in head and neck cancer incidences, even if we go to 100% vaccination rate today, those patients who will be getting cancer over the next 20 to 30 years are already infected with the virus, right? And so it's going to take another 30 to 40 years before we start to see that decline in the number of incidences of head and neck cancer. Right? So a significant unmet medical need that has to be addressed. And we believe that with this Versamune technology and the data we've shown so far in our phase 2 human clinical trials, we've initiated the phase 3 human clinical trial, that we have significant potential to address this growing medical problem.
And speaking of significant unmet medical needs, obviously the HPV program, the HPV Versamune program is the lead asset, but you have many other things going on. A program that's where the sort of the neoantigen package targets something called T-cell receptor gamma alternate reading protein, or TARP, AML, prostate cancer, breast cancer, highly elevated, and then the mucin-1, which is something we talked about actually on a previous show, this interesting glycoprotein that gets overexpressed. Here you're talking about ovarian, breast. Clearly, the ability to mix and match these various neoantigen packages in a Versamune technology. Talk us about some of these earlier state programs as well, because you're much more than the HPV program.
That is correct. So I think one of the key things that we have done over the last several years is get to the point where we have enough substantial data. We recently had the cervical cancer data published by MD Anderson in Clinical Cancer Research. The data was published by the National Cancer Institute in the study we did in JAMA Oncology. We now have these peer-reviewed publications we now have this substantial data that has stayed consistent across 3 phase 2 human clinical trials. Right now we've moved in phase 3. So we have really strong evidence that this technology appears to be working in overcoming some of these limitations. So you talk about the various programs we have. So we have the cervical cancer program beyond the head and neck cancer program, as you mentioned. This specific asset itself, the Versamune HPV, is broadly applicable to a wide range of HPV cancers that are increasing: anal cancer, cervical cancer, vaginal, vulva, penile cancers. This specific asset is applicable broadly to all these cancers. So a huge market opportunity for this asset itself. But beyond that, you mentioned, for example, the MUC1. MUC1 is a protein that is broadly expressed in multiple solid tumors, right? And what we have shown is that by taking that MUC1 protein and pairing it with our Versamune platform, we can generate similar levels of T-cell response. Right? We have filed the IND for that program, and that should be hopefully moving into clinical trials in the near future. It's being done in collaboration with the National Cancer Institute. So that's going to be run and led by the National Cancer Institute. But we also have the PDS-01 ADC, our IL-12 antibody drug conjugate, that's been evaluated in a number of phase 2 clinical trials. So, for example, we have the hepatic artery infusion pump, which is utilized to treat a number of cancers like colon cancer, cholangiocarcinoma, for example. That induces necrosis in these tumors pretty effectively. And that's ideal for our IL-12 antibody drug conjugate because it targets necrosis, it targets free DNA. And so that trial is currently ongoing in colorectal cancer, phase 2 clinical trial that the National Cancer Institute is very optimistic about. And then we also have 2 trials going on in prostate cancer. So enzalutamide, owned by Astellas, is the standard of care for biochemically recurrent prostate cancer.
Yep.
Right. And so there's a phase 2 clinical trial combining enzalutamide with our IL-12 antibody drug conjugate versus the enzalutamide alone. That's also going on at the National Cancer Institute. And there's a 3rd trial that's being run in castration-sensitive and castration-resistant metastatic prostate cancer. And that is a combination of this IL-12 antibody drug conjugate with docetaxel. Right. And so that's combination. So we can see these combinations with standard of care. And so we have a number of clinical trials running currently, phase 2 clinical trials. We anticipate that we should have some data readouts from some of these trials ongoing at the National Cancer Institute anywhere between 6 and 12 months from now. And I did mention that the phase 3 clinical trial in recurrent metastatic head and neck cancer, that's already underway in recruiting patients. And so we have, as you mentioned, a number of opportunities in applying our 2 platforms, both the Versamune and the IL-12 antibody drug conjugate, in a number of cancers that desperately require some improved therapies and improved patient survival.
Mm-hmm. And continuing along that, Frank, and I know you're first and foremost an oncology company, but I just got to ask, because when you go down in the pipeline, we come to something called PDS-0202, which is not part of the Versamune platform, but something called Infectimune, which looks like a key or a doorway to a universal flu vaccine. The pan-flu concept is something we have touched on previous shows, but would just whatever you can say. I know this is preclinical, but love to hear about how you think about the general platform in sort of one and done potential flu vaccines in the future. Love to hear this, whatever you can say about it.
Well, the universal flu vaccine is not something we talk about very much. This is actually work that's being done using our technology and being led by the NIAID. So this is a program that the NIAID is leading, but it's a very interesting program. There's a lot of promise over here, and we're working very closely with Dr. Ted Ross,
Sure.
who is at the Cleveland Clinic, one of the leaders in the field of influenza, and also with another professor, Dr. Andrea Sant at the University of Rochester School of Medicine. Right, she's also working with the NIAID on this concept. But the key here is Dr. Ted Ross, before AI became a big thing, his approach to universal flu was, if I can take all those universal flu strains that have occurred over the last 20 years and understanding how those have continued to evolve and show up every now and then, right? Can I develop can I take a computer-assisted approach to designing a protein that contains the antigenic regions, or the portions of those viruses that the immune system could recognize over the last 20 or so years, and build it into one protein that I could then use as a vaccine to actually activate the immune system to detect and protect against a broad range of viruses. So that was his concept. It was a great concept. However, there's something else that we know we need to generate a really robust immune response. Vaccines today have focused on antibodies, neutralizing antibodies, right? And neutralizing antibodies are very specific to the areas of the viruses that change. Those portions of the virus evolve because the viruses know that that's how they can evade neutralization by the immune system. So those regions that we can generate those neutralizing antibodies against are constantly evolving. However, if you can generate T-cell responses like we've shown our technology can do, you can generate T-cell responses against the conserved regions of the viruses, those regions of the viruses that don't evolve and change from strain to strain. Yeah. And so taking these two concepts together, taking these COBRA computer optimized proteins together with our Versamune technology, right, where we can now address both sides together. We can generate broadly reactive antibodies across multiple strains, but not only that, we can now also generate powerful T-cell responses against multiple strains of the virus. And so that concept is now being tested in animal models. So they've reported some ferret studies at Immunology 2025 a few months ago. And what they have shown in the ferret models, which are the models that you go into just before you go into humans, is that this approach can actually generate very broadly neutralizing antibodies. And it protected the animals against every strain of flu that was used in a challenge, right? And so typically you would vaccinate them to protect the animals, and then you challenge them with high quantities of the specific virus and show that they are protected. So in principle, in preclinical studies, this approach appears to be working, right? And so the next step would then be to take it into a human clinical trial. And so we're also waiting to hear back from the NIAID in terms of what their timeline is and what their plans are. But this would get us to a point where we are not always behind the 8 ball trying to figure out what strain of virus is going to be dominant in any particular season and rushing to develop a vaccine for that strain and finding out that the vaccine was only 30% effective for a particular year, right? You would have a vaccine that's broadly protective and that could be effective year after year, and hopefully also provide more durable response, especially when it comes to the elderly, right? That's one of the major issues with influenza vaccines. The most impacted are the very young and the elderly, who don't generate really good, robust immune responses to even the current vaccines, right? And with this approach, we could potentially generate much more robust and broader immune responses against flu that could be highly beneficial, especially in the elderly population. So that in a nutshell is what's going on with the universal flu vaccine.
Yeah, no, again, I appreciate you going into what you could go into with it. I just, again, I saw it there, and it's just another sort of impressive piece of how you think at the company and sort of the potential repurposing, let's say, of these really elegant tools that you put together. Frank, what else is coming up that we should know about while we have you today? Clearly a lot in the late stage of the clinic now, and that's very exciting. Anything else as we get into the later part of 2025, get ready for 2026 that I missed that you want to mention? Other conferences, talks, places that we could run into and meet you at some point? Anything else while we have you today, please?
Right. No, I think some of the key things, some you mentioned. First of all, the fact that the phase 3 clinical trial in recurrent metastatic head and neck cancer has been initiated in the most prevalent type and rapidly growing type of head and neck cancer today. So we see huge opportunity over here. We will give updates as we go along in terms of the recruitment and also the interim data readouts. We hope to have some interim data readouts from that trial as recruitment progresses. Very, very importantly, also, you did mention, we talked about the prostate cancer program ongoing at the National Cancer Institute. We expect that the principal investigators at the National Cancer Institute will very likely be attending some conferences, some of the big conferences in the near future. And we'll provide updates on where those conferences are and who will be presenting that data at the upcoming conferences. The same with the colon cancer program that's also ongoing at the National Cancer Institute. We expect to provide updates at upcoming conferences. And with the head and neck cancer program, we anticipate that we could potentially be presenting at some of the major oncology conferences also coming up later this year. But we also anticipate that we will also be presenting updates on the final updates on the head and neck cancer trial, the phase 2 clinical trial. So even though we've started the phase 3 human clinical trial, the phase 2 hasn't been completely wrapped up yet, right? We have the last patient who was dosed not too long ago. And so we will be providing the final data readouts from that trial also, as well as the final data readout from the MD Anderson trial in cervical cancer. So a lot of updates that we would like the community to be aware of. We hope to be able to provide these updates as we progress development of these immunotherapies. But I think we are very optimistic that this next generation cancer vaccine technology could potentially overcome a lot of the hurdles we've seen with CAR-T and also with the cancer vaccine approaches. And the data we've generated today makes us extremely optimistic about the potential, not just in the head and neck cancer, not just in the HPV cancers, but all these other tumor types that we've talked about today.
Absolutely. Yeah, it's an exciting, it's an elegant story. And I neglected to mention too at the beginning of the show when I was researching you that your PhD, which you did 30 years ago, which was on these long circulating liposomes, now comes full circle with these nanoparticles. So just as a point out that you followed your passion and throughout your career, and it's again, biotech takes a long time, but I think
it takes a long time.
Yeah.
No, it's interesting because I credit Professor Leaf Wang. Professor Leaf Wang is actually the scientific founder. He used to be my PhD thesis advisor several years ago at the University of Pittsburgh School of Medicine. And he was actually the one who convinced me to leave and help him with PDS and get PDS started. But he actually came up with that initial concept. So he definitely deserves a lot of credit for coming up with the idea and actually insisting that I leave KBI to help him get this done. So
it's funny because he mentioned your PhD in his bio, and it's like, whoa, 30 years, but still, it's nice to see this all come together this way. But no, very impressive. And I look forward to continue to follow you and the company as you get close to registration here. For everybody again that's going to be listening to this episode of the show across the various podcast networks. Or watching on the YouTube channel. You've been spending time with Dr. Frank Bedu-Addo, President, Director, Chief Executive Officer, PDS Biotech, doing really amazing things to usher in this new generation of targeted immuno-oncology treatments. Frank, again, thank you for taking the time out of your schedule to talk to us for a little while today about everything you have going on. Thank you for everything you're doing for the patients out there. And as we like to say on our show, thanks for helping to create a better tomorrow for so many people via the type of work you've been championing. Very, very interesting story.
No, Ira, thank you very much. Thank you for all the work you're doing to make some of these better understood in the community, and also thank you very much for the opportunity to chat with you today. Really appreciate it.
Really great having you.